Welcome to AP Biology. Today I want to talk to you about viruses. So the next slide is of a pathogen that is not a virus. This picture is a bacterial cell with a bunch of viruses, bacteriophages on it. So the next picture is a tapeworm, but I want to talk to you about it because I want to define pathogen because viruses are pathogens. So a pathogen is a biological agent that causes disease in its host. A parasite, very similar word, is an organism that lives in or on another organism for part or all of its life cycle. So the host may or may not get sick. So for example, head lice is a parasite. I'm not so sure that you'd call it a pathogen. I'm not really sure that it causes a disease. It can cause other stuff to infect your scalp if there's too much of your skin that's getting cut up. Okay, another vocab word, host. This is the organism that is being infected. And while we're at it, infection. An infection is when the body gets invaded by something that causes disease, like a virus or bacteria, or it could be a fungus. So sometimes students mess up the word infect with effect or affect. I don't really care if you know the difference between these. We've got a noun and a verb, but infect is different. So the effect of something on something else, like the effect of the weather on my mood, right? That's how you would use this term. Or the weather affects my mood, right? But infect means that there's something inside you growing. This is a different word. Okay, so virus examples. This is what HIV looks like, the virus that causes AIDS. This is what the EBOLA virus looks like. This is what the herpes virus looks like. Adenovirus, influenza virus, papillomavirus, bacteriophage, any bacterium that infects, any virus that infects a bacterium is called a bacteriophage or phage, and they kind of look so cool, don't they? Pandora virus is huge, and it has more DNA than some bacteria, which is, to me, just the coolest thing ever. Okay, so viruses are not cells, so some people will define them as not alive. To me, I don't really care if you define them as alive or not alive. What I want you to do is I want you to know how they affect living things. So a virus is a very small infectious particle. It consists of a nucleic acid, DNA or RNA, right? So nucleic acid, enclosed in a protein coat, and in some cases there's a membranous envelope around it. So when we define life, we say all living things on this planet have DNA as their genetic material. So if you define that as life, I guess a virus wouldn't be alive. But viruses have some genetic material. So I guess who cares which one it is? But interesting that it can be either one, right? Okay, so viral genomes can consist of either double-stranded DNA or double-stranded RNA or single-stranded DNA or single-stranded RNA, which is weird, right? In everything else, you always hear about double-stranded DNA, right? But viruses can have either double-stranded DNA like we have, with the A's matching the T's and the C's matching the G's, or it can have single-stranded DNA, or it can have double-stranded RNA or single-stranded RNA. So how would you know? If it has A's, T's, C's, and G's, it's going to be DNA. If it has A's, U's, C's, and G's, it's going to be RNA. If the number of T's and A's match each other, it's probably double-stranded. If they don't, then it's not double-stranded. Again, like or C's and G's would have to match, right? For RNA, if A and U matches, like if 20% of the sample of virus DNA is adenine and 20% is uracil, they're probably matching up in a double helix form, because that would be a really weird thing. Like as long as these are the same, right? So that would be 30 and 30% here, right? So if you see those kinds of ratios, it's probably double-stranded. Whereas if it says like the AUCG, if you get like 20% of adenine and 35% uracil and 30% cytosine, what do we have? 30, 85, so it's 15, right? 95% G. If you see ratios like that where the A's and the U's don't match and the C's and the G's don't match, it's probably single-stranded. A capsid is the protein coat that encloses the viral genome, so they can look different. The really funky looking ones here are the viruses that infect bacteria. Some viruses also have membranous envelopes. Usually those come from the host that they're like sort of coming out of, and they're often going to help the envelope infect the host, or help the virus infect the host. These are often how viruses in animals look. So viral envelopes are derived from the host cell's membrane. They're going to contain the host's proteins and stuff on the outside. So like if this is the host cell and here's the virus that's budding out, it's going to have proteins and carbohydrates on the surface that belong to the original cell, right? But then the virus takes over and it will also make some proteins that will go to the cell surface that help it to infect another cell. So the viral envelopes, those membranes, they derive or they come from the host cell membrane, and they contain a combination of viral and host cell molecules like proteins and carbohydrates. Okay, viral specificity, this just means that viruses are very specific. So a virus that infects a person usually doesn't infect a cat, but there are some crossovers. That's how we get new viruses in a human population, right? Maybe the virus came from a bat or some other place. So most viruses infect only specific hosts or host range. So viruses that infect humans usually don't infect cats, though there are certainly some exceptions. Did I freeze? Okay. General features of viral reproduction. Viruses can replicate only within host cells. This is really important. They cannot replicate alone because they don't have everything they need to replicate. So they don't have ribosomes for making their proteins. They don't have all the enzymes needed for making RNA from DNA or whatever it needs to do. So typically what happens is the virus gets into the cell. If it's made out of DNA, if the virus is made out of DNA, then it's going to use the enzymes in the cell already to replicate itself. Sometimes there's an enzyme or two present in the virus, but they're going to need some of the enzymes from the host. And then you'll use the enzymes also that are in the host cell to go from DNA to RNA, to make the RNA. And then they're going to use the ribosomes that are in the host cell to make their proteins. Then they'll assemble. So once a viral genome has entered the cell begins to manufacture the viral proteins. The viruses make use of the host's enzymes, the host's ribosomes, the host's transfer RNA, the amino acids present there, any ATP that it needs, other molecules, etc., etc. So the virus has the instructions. The virus has the genetic material to reproduce. It just doesn't have all the materials it needs to reproduce. It doesn't have the ribosomes, all the enzymes. Viral nucleic acid molecules and proteins spontaneously self-assemble. Here they are getting together, and then they either bud out like they're doing here of the cell, sort of a type of almost, I don't know, it's sort of like exocytosis, but a little bit different. So it's budding out, and I guess it's exocytosis. Shouldn't have said that. Exocytosis, you're like, blah, releasing stuff. This is more of like a whole vesicle that's leaving. Or sometimes if it's like a virus infecting a bacterial cell, they might just break the cell open and destroy it that way. So bacteriophages, or for short just phages, sometimes you'll hear phage, these are viruses that infect bacteria. They are really well understood because it's easy to grow bacteria, and so it's easy to study these viruses. And they look the coolest. So a protein tail piece attaches the phage to the host, and it injects its DNA. So this is the DNA getting injected, or it could be RNA. Phages have two reproductive mechanisms, either the lytic cycle or the lysogenic cycle. Lytic cycle is a shorter word, and the cycle is actually quicker. So the virus will attach over here to the bacterial cell. It only fits onto certain cells, so it has to have the right receptors. The receptors are made for something else, but the virus just takes advantage of that, sends in its DNA or RNA, and then this DNA goes to work right away. It uses the cell's enzymes to make RNA, and then it uses the cell's ribosomes to make proteins, and then they self-assemble and they kill the cell, and that is called the lytic cycle because it lyses. Boom, cell dies. Bye, cell. All those viruses, they go out and infect something else. Here's the lytic cycle. You just pretty much kill the cell right away. The lytic cycle ends with the death of the host cell. Produces a bunch of new phages that break open the host cell, releasing all these new viruses. This is the more virulent it is, the more likely it is to kill you. So the phage reproduces only by the lytic cycle. It's called a virulent phage, and the more virulent, the worse it is. Okay. Bacteria have defenses against phages called restriction enzymes, which we'll talk in the biotechnology unit. We'll talk about it a little bit. But here is a bacterial cell. And some of them have this restriction enzyme. So here's a restriction enzyme. And if it recognizes the DNA that's not part of its own, right? If it recognizes it might be in its own, but it would be methylated. If it recognizes a particular site, then it will cut the DNA. And so if this viral DNA has the particular code, like maybe the restriction site, maybe it goes AA, CC, GG, TT, in which case the other side would be AA, CC, GG, TT. Anyway, if it recognizes this particular site, maybe it cuts it between the C's, it's going to cut that viral DNA, and that will save it. That will save the bacteria so it can live another day because it kills the viral DNA once it's inside, or maybe the viral RNA, whichever it is. So these restriction enzymes are really cool because they're something that the bacteria uses to save itself, to kill viruses, but it's also something that scientists can use, which is kind of cool. Lysogenic cycle is when the virus infects the bacterium, but it doesn't kill the bacterium right away. So the beginning's the same. It has to be specific. The virus has to attach to the particular receptors on the surface of the cell. Remember, the cell didn't make that on purpose. They just happened to be there. It injects its it injects its DNA. The DNA doesn't go ahead and make RNA and proteins right away, though. Instead, it inserts itself into the bacterial chromosome, at which case you're going to call it a prophage. That just means it could be bad, but it's not right now. So it's like a future virus. At some point, there's a trigger that will allow the DNA the DNA to come out and then do the whole lytic cycle thing and eventually break the cell. So lysogenic has a longer word, and the bacteria lives longer after it's infected. Okay, so here's the thing with this one. As the virus injects its genetic material, the genetic material inserts itself into the host chromosome. So this is the host or the bacterial's chromosome. And then the thing that the last video didn't show very well is that this cell can then go ahead and reproduce. And so here are two new bacterial cells. They just reproduced by binary fission, and they both have that little bit of virus DNA that we're going to call a prophage now. So the bacteria can reproduce over and over and over and over again like this, and then there might be some trigger at which time the viral DNA will come out of the host chromosome, and it will go into the lytic cycle and kill the cells. So the lysogenic cycle replicates the phage genome without destroying the host for a while. The integrated viral DNA is called a prophage, this little piece right here, from here to here. Every time the host cell divides, it copies the phage DNA and passes it on to new daughter cells. Here are some new daughter cells that have the viral DNA in them. Some environmental signal can trigger the virus genome to go out of the lysogenic cycle into the lytic cycle. So this little piece here gets itself out and then copies itself and makes some RNA, which makes some proteins on a ribosome, which then makes a whole bunch of viruses, which then assemble themselves and break the cell, and they all die. So these are called temperate phages because they allow the cell to live for a while. All right, so Horizontal gene transfer, we talked about that before. There's transduction, and that's when a virus is packaged with a little bit of host chromosomes. So here's a new virus, but it has some bacteria DNA in it instead, and that is now going on to a new bacterial cell. So we talked about horizontal gene transfer before. There's transformation, transduction, and conjugation. And so today's was, I just quickly went over transduction with you. Okay, so in viruses that infect animals, there are a couple of different variables used to classify these. Are they made out of DNA or RNA, and are they single-stranded or double-stranded? And so here are just some examples. You don't need to know any of the examples truly, but we will talk a little bit about retroviruses. Right here, you'll need to know a little bit about them. And then curiously, look at this. Here's the coronavirus, which I've taught this unit a gazillion times and never noticed it before. You know, heard of SARS, right? That was interesting, but not in this area of the world. And wow, now I know all about the coronavirus. All right, so viral envelopes. So the different parts of the viruses. Many viruses that infect animal cells have this envelope that came from the host that it infected last time to help it get into the new cell. Viral glycoproteins, that's carbohydrate attached to a protein on the envelope, bind specifically to some receptor. So this is just an example. It's the XCT receptor, and this glycoprotein attaches to it. If you have a mutation in this receptor, it's possible that that virus can't infect you. So sometimes mutations are good. Some viral envelopes are formed from the host plasma membrane as the viral capsid exits. So here's an example where the virus, here's the viral DNA, the red part, and then the protein around it, and then it gets some of the membrane from the host cell. The broadest variety of RNA genomes is found in viruses that infect animals. Retrovirus is a type of RNA virus. It's not the only one. So you need to know that some viral genetic material is DNA, other viruses have RNA, and AP does ask questions on retroviruses frequently enough. So a retrovirus, think like retro backwards sort of. So the normal way that genetic information flows is that you have DNA, and that DNA codes for RNA, and if it's messenger RNA, then that codes for proteins. So retroviruses aren't like that. You start out with RNA, and then you have an enzyme called reverse transcriptase. ACE, that's an enzyme. Transcription is DNA to RNA. Transcription. RNA to protein is translation, which we'll talk about soon. Translation. Okay, so transcription is DNA to RNA, so reverse transcription must be RNA to DNA. So there's this enzyme called reverse transcriptase, and that will change the viral RNA to DNA, and then the RNA will get destroyed, and then DNA polymerase can make this double-stranded DNA. So it flows backwards. You don't go DNA to RNA like we always do. Instead, you go RNA to DNA, and then DNA to RNA, and then RNA to protein. So this part is normal. This part is the different part. So this DNA can insert itself into the nuclear chromosome, and then from there, you can make the RNA and then make the proteins, right? So here's some RNA that's made. The RNA can also make the proteins needed, including this enzyme. This reverse transcriptase isn't in animal cells. That's part of the original, it's one of the proteins that's in the original virus. So retroviruses use reverse transcriptase, it's an enzyme, to copy their RNA genome into DNA.